Good morning, good afternoon, good evening to everyone joining us from all around the world. Welcome to Viva Bio Insight. For those who join us for the first time, Viva Bio Insights is a flagship event hosted by Viva Biotech. We established this series to foster dialogue on important topics and to collectively facilitate and accelerate global bio innovations. My name is Bing. I'm currently Vice President and Head of Early Discovery Platforms at Viva Biotech. I'm your host today. I won't read the slides, but take-home message is I was trained as a photoorganic chemist at Boston University. My expertise is DNA encoded library technology with about 20 years experience, starting out from GSK. Actually, I'm based in Boston, and my passion is to help Viva to be the best screen center in the world. Okay, as always, Viva BioInsights has another truly exciting session titled AI times flow chemistry from bench to business, academic, and industry perspectives. This is the agenda. We'll give the more detailed introduction of our speakers, and we'll reserve 20 minutes in the end for Q&As. All questions are welcome. Please feel free to submit them to the Q&A box. Also, as usual, before we move into the formal presentations, we'd like to thank our sponsor, Viva Biotech, and also we'll spend about 2 or 3 minutes to share the evolution of Viva Biotech and our vision of the future. Okay, the history and the milestones of Viva Biotech can roughly divide into 3 phases. Phase 1 is foundation phase. We founded our company around 2008, 2009 with protein structure and bioassay platforms. By 2012, we set up our very strong hit discovery platforms such as ASMS, SPR, GPCR member protein technologies. And by 2016, we set up Viva libraries. And the second phase, we called it growth phase. So as you can see, there are a lot of milestones here. You know, we listed in Hong Kong, IPO 2019, then 2028, 2020, we purchased Langhua, then we expanded our service chain to the CMC and CDMO. 2021 acquired Synthesis. 2020, we moved to this new headquarters. Then phase 3, we called it accelerated expansions phases. As you can see, a quite dense list here. We expanded the XDC, DNA encoded library technology, molecular glue, new modalities such as peptide drugs. And now we have, alongside with very modernized ADD and CDD. To support this, to support this innovation scale, we actually have built up a strong global network. Our footprint covers almost all the major biohubs across the world. For example, in China, we had in Shanghai, we have Suzhou, Jiaxing, Ningbo, Taizhou, Chengdu. Those facilities, actually, we have about, accumulatively about used square feet is up about 1.2 million square feet. And also global footprint, including Boston, San Diego, London, Belgium. Actually, we also have San Francisco. To back up this, global reach, we have a team about 2,100 professionals, including about 1,200 scientists, more than 50% of them hold master's and PhD degree. So taken together, so Viva has already evolved to a highly integrated research partner across discovery, development, and manufacturing. So to helping our clients to move their projects forward with great success and efficiency. Hopefully we have a chance to collaborate. Okay, switch gear a little bit back here again. So it's my great pleasure to introduce our first speaker, Professor Jie Wu from National University of Singapore. Jie obtained his PhD in 2012 with Professor James Panic at Boston University, working on natural productive synthesis. Actually, that's about 20 years before. That's why Jie and I met there. He's very talented, one of our best graduate students at that time, rising star. And then he moved to the MIT with Professor Tim Jimson and Professor Alan Hanton. Jie has posed a hardcore continuous flow chemistry. He's one of these pioneers and the major players in this field. Since joining the NUS in July 2015, his research group focused on new synthetic methodology development using photocatalysis assisted by advanced flow technology. His group is also interested in the development of advanced flow technology for on-demand and automated synthesis of functionalized organic molecules. We'll hear more today. In July 2012 or 21, Jie was promoted to tenure associate professor. I had a chat with him. If everything works smoothly, we're talking about he's going to be a full professor, and not in months, it's almost maybe in weeks. So congratulations. And he's a recipient of a long list of the awards, such as Dean's Chair Professor, Tokyo Chemical Industry Award, NUS Young Research Award, the Same Chemistry Journal Award, you name it. So without further ado, welcome again. The floor is yours.
Okay, thanks, Bean, first for the kind invitation, and it's my great pleasure to be here, and also say old friend. It actually bring me back to like 20 years ago, which I met Bean in Boston University. Even though we are in different groups, but we are very close, as he is always willing to help others. So it's my great honor and pleasure to be here today to join this symposium. So I would like to give you a little bit introduction about what we have done in National University of Singapore last 10 years. And just now been told me I need to speed up a little bit because there's too many slides. So if I speak too much, too quick, please let me know after my presentation. So I think I don't have to introduce too much concept about the flow technology, as it is quite popular in industry for scaling up or for any dangerous reactions. So flow system is different from a batch flask, which we use pump as a driving force. And then they were pumping the reagent to the tubings and the reaction starts. And then you're adding quenching, and then on the other side, you get the product. So it's continuous production. It's like a Lego. You can put a specific reaction apparatus for your own specific reaction. And it has many advantages, such as enhanced mixing efficiency, so you can do heterogeneous reaction more efficiently, and also very good heating mixing efficiency, so you can run high T, high P reaction. Photon efficiency is also good. So most of the photon reactions in flow, you can have light penetration very efficiently and uniformly. And the safety and scaling up is one of the best thing in flow. So that's why it's very popular in industry. Like a reactor like this table size, it can actually give you tons reaction. But when we build up the group, we look at the problems in flow. So hopefully we can solve some of them. in order to help the synthetic community to use them better. So for example, in flow, the solid is a problem because it causes clogging in the microtubings. And the slow reactions, normally we don't run in flow. Normally the reaction time is less than an hour. If the reaction time is too long, then you need a very long tubing and the throughput is very low. And the multi-step synthesis is always a problem in flow if we really want to do an end-to-end synthesis because the reagent solvent compatibility is an issue. Another interesting thing in flow is we think if we have something built in lab, we can directly scaling them up to the industry scale. And that's one of the attracting point about the flow chemistry. In my lab, we also like flow because they are easy to do automation. So for example, we only need to control the pumps and valves. We can do the automation. Not like the batch system, you need a lot of controlling parameter in order to achieve the automation. So in the past 10 years, we actually developed several flow technologies based from a chemist angle to solve some real problems when we're trying to do research. So I will introduce some example to you today. So first of them is we build a so-called star flow. So the star flow is actually a batch reactor. The difference is we add the two tubings before and after, two valves before and after the tubing. So those valves have two functions. One function is they can hold pressure. The second function is they can taking off. So actually, after we're taking them off from the flow system, it becomes those microtubing reactors. So you can consider them as a batch reaction. You just change the formology from round bottom flask to microtubings. But when you change them, there are some benefits. That is, the light penetration is better and they can hold pressure. So we target we think we have a good system to target those photo reaction under high pressure. Because before, when you do some photo reaction under high pressure, you need this very tedious or very difficult high pressure gauge and with special glassware and a special light source. But now we just need those tubing and two valves. You can do this reaction even more efficiently. So with this, we actually did a lot of gases involved transformations, like those natural gas. For example, the alkene, CO2, freon, ethylene, acetylene. We can just utilize them to incorporate some value-added compounds using those natural gases. So just now you can see we have this star flow microtubing reaction. This reactor, everything is static. So the mixing is actually achieved by diffusion. So the mixing is actually pretty slow. So we target on slow reactions. So how about we push to another angle or push to another extreme? That is, we can flow them as quick as possible. So flow as quick as possible, then you cannot do continuous flow. Then we have to do the circulated flow. So continuous flow is from A to B. But the circulation flow is from the bottle A back to the bottle A. And until the reaction finishes, then you get the region out. So you can think of this as a batch reaction as well. But now we flow them really quickly. So in continuous flow, we normally flow as less than 1 ml per minute. But here we can flow in my research lab, we flow more than 300 ml per minute. So with this very high flow rate, we actually can flow solid. And it also naturally gives us the turbulence flow. So when a chemist do reactions, we have very strong stirring. The stirring is trying to generate a turbulence flow. But in those microtubing reaction, it naturally generates turbulence flow. So give us a very high mixing efficiency. And now we can tolerate the solid. And with this, my student Chen Guang actually developed this flow system. You can see the reactor is actually pretty small. It's just half fume hood size. And we can run this reaction, this photo cross-coupling reaction in 100 gram scale. So this reaction is published by Professor Koenig in a Nature paper. They use MPGCN as a heterogeneous catalyst with nickel catalyst. So it's a Backward type coupling, but without a ligand. It's under photo condition, and you can see that if we run this reaction at 100 gram scale, the efficiency is about 10 times compared to the batch reaction. And for the cis coupling, we also can get 100 gram scale. So this is another example we steal from the science paper, which is a three-phase reaction. The trifluoromethylation reaction, we can have the air as a, because it's oxidative coupling, so we use air as an oxidant. MTG7 is still heterogeneous, so it's a gas, solid, liquid, three-phase photochemical conditions, need a little bit of heating. So you can see this condition is actually pretty complicated, right? If we want to run this in batch conditions, it's actually difficult to control. And the reaction has been published in the paper with 20 milligram with 45% of yield. And now we can achieve this reaction by 10 times circulation. So it's a semi-continuous production with 210 hours, just with a half size of the fuel hood. We can get a kilogram scale, and the yield is almost the same. So that means now we have probably we have a system. If you have a reaction which you get a good yield at a 20 milligram scale, and then we can scaling this up to kilogram for you. Just use our simple high-speed circulation flow system. And then we think we have a good system, we can do some heterogeneous reaction. So we developed some heterogeneous catalyst. And the first catalyst we developed is a COF, because we like COF, covalent organic framework, because COF can incorporate different catalytic system to the same one. So that means we can do some multifunctional catalyst. And the catalyst we interested is propylitrodrin's two-in-one system. So in this system, the developable photocatalyst, which behave a photodie, and also it's a cross-coupling reagent. So then we put those catalysts in a COF, and then this is how we generated the COF, and we actually did some characterization to understand the nickel is the nickel 2. And then you can see this COF catalyst can actually be applied to a variety of coupling reactions. It actually can do like a CS, CN, OCC, CPCS, C boron, C chloride coupling. It's one of the most versatile catalysts I have ever seen for the cross coupling. And you just need a little bit base and this heterogeneous catalyst, and you can recycle them for 10 times without any problem. So the reaction is pretty mild and use a visible light. And also the scope is really broad. We can, and when we compare to propylitrodrin's homogeneous catalysts, we actually found that our catalysts give much higher yield in most of the case. And the reason is that in their photocatalyst condition, they use UV light. But when we make this catalyst to COF, it goes to redshift and it goes to the visible light. And the visible light will not induce dehalogenation byproduct, so we get a higher yield. And then we can simply use our high-speed circulation flow system to achieve a decagram scale reaction. We didn't do too large scale. The reason is that in academic, we have limited resources and space. But if you want to run this reaction in kilogram, I don't think it's any problem. And the second reaction, we actually recently interested in the single atom photocatalyst. So single atom catalyst is recently a very popular topic, which pioneered by Professor Zhang Tao from China. So he may win Nobel Prize if one day the single atom catalyst was being recognized by the community. So in this system, we actually targeted in the dehydrogenative cross-coupling reactions. So CDC reaction, everyone knows, is one of the green reactions, but you normally need some oxidant because of oxidative coupling. So if we can generate hydrogen in this reaction, then we don't have to use external oxidant. So how we do this is we collaborate with our colleague Prof. Lu Jun. They help us to make some single atom catalysts with platinum. So we know that the PCN is undergoing photoredox condition, and the platinum can generate hydrogen from proton source. So then we just put this heteroaryne with nucleophile, and then we will generate this coupling product with hydrogen generated as a byproduct, which then we don't need oxidant. So you can see this reaction tolerates with a variety of heterocycles, as well as different nucleophiles. So even the acid can be coupled in this case. So it's a decoupling mini-skid type reaction. So the byproduct is only CO2 and hydrogen. So it's actually very, very green reaction. So we can utilize them to generate some biomolecules as well as some function molecules. And we can use our high-speed circulation flow system to achieve a 10-gram scale reaction without any problem. The next example I want to show you is a gas-involved transformation, which we are typically interested in this ethylene difunctionalization. So this is one of the type which have arene with two methylene groups with a heteroarene to generate a three component coupling reactions. And the coupling actually can be achieved by like use ethylene arene and heteroarene. That's the best way to couple them. And it's very modulated. But to achieve ethylene difunctionalization is actually very difficult because the ethylene can undergo polymerization and the reactivity with radical is actually pretty slow. And at the same time, it's a three-component reaction, so any two components will actually become a byproduct. So we did this reaction to borrow the chemistry from Tobias Ritter that uses thyrenium salts, which we can generate radicals from using the photo redox conditions. And then the radical generated from arene is a sp2 radical, which is electrophilic. So it will favor the electron-rich alkene instead of the electron-deficient heterine. Then generate the sp3 radical, which is nucleophilic. Then it will react with a electron-deficient heterarene. So because of this polar match, we can generate selectively this three-component cross-coupling product. And the interesting thing is that you can directly use ethylene to link the two drug molecules together. That's actually quite interesting for this kind of reaction. And we can also generate some derivatives from this compound. So here I want to show you a close picture of how we do this high-speed circulation flow system. So you can see it's actually pretty simple, right? So this reaction is ethylene. We use 3.8 atm, so it's a little bit high pressure. And then we use thyrenium salts, which is not soluble in the solvent. So it's again is a gas solid liquid three-phase reaction. And you can see the setup in the middle is a reactor and it's flowing quickly. Okay, you can see that it's a gas, liquid, solid. It's three-phase slug flow. And the mixing efficiency is really good. And there is a reservoir. So the reservoir at the bottom, it's a slurry. And then at the top is a gas. So we have two pumps. One pump pumping the gas, one pump pumping the slurry. And then you can think about that. The gas will actually react at a high pressure. But if it's not finished, then the gas goes to the reservoir, and by weight, it will separate. By gravity, it will be separated from the slurry. So that means you can just use a very small amount of gas to achieve high pressure and until the gas was finished. So in order to achieve high pressure gas involved reaction, you don't need a lot of gas in the future. You just need a balloon or a little bit of gas to achieve this condition. So it's actually a very good system for the gas involved reactions and also very good system for photo involved reactions. So based on this, I will move my talk to the on-demand synthesis. So one of our goals is in the future whether we can have a small molecule synthesizer that we push a button, we can make a molecule. So that's our dream, and there's a lot of chemists' dream. But as we know, all the organic molecules grow differently. It's like a human; they have different characters. They need different conditions or reactions to achieve them. And this map actually shows you the state of art of the automated synthesis. So actually, there are two directions. One direction is a target-oriented synthesis. That means we want to make a specific target in a large scale. So the representative work is from Klaus Jensen, Tim Jamison, Lee Cronin's computer system, and Andrew Cooper's robotic chemist. And another direction is a diversity-oriented synthesis. So diversity-oriented synthesis is you target at the small molecules, but a large quantity of different structures for improve the function of the molecule. And currently, not too many groups working on in this area. I will talk a little bit more why. But one of the representative work is from UIUC's Martin Berg's group. They use this block CC chemistry, which is use Suzuki coupling to generate a Manda boron, and then to mimic the amide coupling. And all the purification was based on the boron reagent. So it's a kind of limitation for the chemistry. But recently, they actually spin off a company called Excelsior Science, and in last year, they raised $95 million to run this chemistry. And our research group actually joined this field by using solid phase synthesis as a unified purification technology. So those are some pioneers in this field, and they all have some companies in this field already. So first, we actually want to use this high-speed circulation flow because we think it has a benefit from both batch and the continuous flow conditions. It tolerates solids and it tolerates slow reactions, which has a problem of the continuous flow synthesis. So then we want to make them to automated multi-step synthesis. And then the problem here is how we can incorporate the different separation technologies to this multi-step synthesis. So first we look at the generality of those reactions, and all of those reactions was done at a gram scale. So with this high-speed circulation flow, we can achieve both heating photoreactions and homogeneous or heterogeneous reactions without any problem. So when we increase this reaction to 100 gram scale, you can see that for the homogeneous and solid liquid reactions, the reaction is almost the same. But when we do the gas liquid reactions at 100 gram scale, the efficiency is much higher compared to the batch conditions. And this reason is the surface area is much better in a flow system. And with any photoreactions, no matter it's heterogeneous or homogeneous, the reaction is better at a large scale. And this is due to the light penetration is more efficient in this tubing reactors. So we think we have a good system to scaling up most of the reactions. And then we apply them to continuous production of drug molecules. And this is an all-in-one system. That means all the reaction. So this is a three-step transformation. It's very classical reactions. The chemistry is simple, but to make this molecule, we use just one reactor and incorporate the different separation technologies. And we can make about 25 gram or 26 gram after 56 hours because it's a three-step reaction. So you can see the system is quite small, it's quite condensed, but the problem is that it's taking too long time. So naturally, we actually applied this reaction to separate the reaction to different reactions. So for example, we have a 4-step reaction, then we have 4 reactors to achieve a semi-continuous fashion. So in this system, we actually incorporated both high-speed circulation flow system and the continuous flow system. So high-speed circulation system will achieve the solid and slow reactions. And the continuous system is still excellent for flash chemistry as well as packed bed catalysis. So with these two flow systems together, we think we have a universal flow system, which we can run most of the reaction in the world. And then compared to the computer system, we can do a large scale synthesis compared to the batch reactions in some difficult reactions. So then we incorporate different separation technology to those two flow systems. And here I want to just show you one of the video we made for this 4-step reactions. And the first 2 reactions is actually solid involve coupling reactions. And the third step is simple deproduction, but it's very quick, just 1 minute. So it's a flash chemistry. So the first 2 we use a high-speed circulation flow. And the third one we use continuous flow. And the last one is a gas involved photoreaction. And it's slow reaction, which needs 24 hours. Then we use the high-speed circulation flow. And the first 3 steps, actually the total reaction time and the quenching time, total together is about 24 hours. And the last step is 24 hours. So we did it semi-continuously. So we accumulate the materials to the last step. And when the last step starts, the first step starts again. So every 24 hours, we can do 1 batch, 1 cycle. I'll give you about 2 gram scale. So now the roadmap started working. And you can see that the system, because we have demonstrated that it can generate a kilogram scale. So if you really want, I think we are very confident to make a kilogram per day production with most of the molecules less than 6 or 7 steps. we can make it directly from the literature to large scale for you. So I think that's the power of this system. So now the third step starts, and most of the thing is actually on the separation part. So within this, and everything is actually doing automatically. So we don't have to have human intervention. The purification was achieved by crystallization most of the time. And the rototherapy is actually used to switch the solvent. And there are membrane separation and also flash chromatography to get rid of some byproducts or impurities. So now you see the light is on, so the last step is on. So when the last step is on, the first step starts again. So it's actually going continuously in a semi-continuous fashion. So I will stop here and save some time. So then I will switch to the diversity-oriented synthesis. So just now you can see we are the universal flow system is actually targeted as a target-oriented automation. But now we're doing diversity-oriented synthesis. So diversity-oriented synthesis, we target a multi-step synthesis because it will give us much larger scope of the derivatives. So you can consider them as a methodology study. So when we normally in academic doing methodology study, we do single step reaction. So when we do single step reaction, we only care how to make those molecules. We don't care about the purification. But when we do multi-step methodology study, we have to care about purification because purification is a problem. And automated multi-step synthesis has already been known by many peptide synthesis, oligomer, RNA, DNA, and Professor Merrifield, when he wins Nobel Prize of solid phase synthesis, he already made the SPS automated. So the problem here, you can clearly see that is a purification technology for all the molecules. So you need to have a verified purification technology for all the intermediates and the product separation in order to do automation. So Martin Berg achieved this by using boron reagents, and we try to achieve this by solid phase synthesis. And this is Martin Berg's block CC chemistry. And we actually collaborate with Prof. Saif Khan at the chemical engineering department. And then we grow the molecule on the solid bits and wash them and grow and grow. The problem here is you have look at the peptide synthesis and the RNA DNA synthesis is simple because iterative synthesis. But for organic molecules, every molecule grew differently. So you need to generate a so-called chemical recipe file, or you can consider them as a multi-step synthesis protocol. So it's like an optimized condition you run in your reaction. So after you generate the optimized condition, you can do the substrate screening. The problem is to generate this protocol is actually pretty time consuming. So this is one protocol we generated. So you can see the yellow one is a reaction, and we have to incorporate the purification, which is a gray one. And the purge one, the pink one is gas purging. So the green one is cleavage. So after we get this optimized condition, we can change the substrate. And recently, we actually worked together with EDDC at Singapore, which is a drug development center. They're actually interested in this, in those antivirus compounds. So those compounds are actually tripeptide. So it's very suitable for solid phase synthesis. So we made those molecules about 13 steps, about 64 hours. And then we upgraded our system to 5 columns per time, and everything was controlled by one computer. So we can make 5 molecules after 60 hours. So we made about 80 compounds for them. And those compounds are all designed by EDDC, not by us. We just behave as a tool and to make those molecules for them. So about 8 of them actually have good nanomolar activity. So 2 of them is further undergoing the mouse study. So another project I want to introduce to you is recently we did something with AI. So previously in the DMTA cycle, we do it linearly. I think it's still one of the best way to do the drug discovery. But the problem is that when we identify a lead molecule, for example, have a good potency, then we need to optimize the toxicity solubility, and etc. But when we optimize toxicity and solubility, the potency may drop. So that means we have to re-optimize the potency again. So it's a linear process, and it's a back-forth campaign. So very time-consuming. So nowadays, people use AI or use data science to optimize the molecule. And when you use AI, the good thing is that you can optimize 10 or more than 10 parameters at the same time. So we can optimize the potency, we can optimize the toxicity, solubility, pharmacology, kinetics, all at the same time. So you don't have to optimize the back-forth. But this gives us another problem when you do a data-driven drug design. That is, the data-driven is really quick because you can generate thousands of molecules by computer within an hour. But to make a molecule, especially multi-step synthesis, it may take you days or even weeks to make one molecule. So there is a big gap between the material flow and the physical material flow. So you can use high throughput synthesis to achieve this problem. But high throughput synthesis, now we know that most of the high throughput synthesis are only doing single step reactions. They cannot do multi-step synthesis. So this is the drawbacks of the make step in the DMTA cycle. So we think, how about we use our so-called multi-step synthesis, diversity-oriented multi-step synthesis in automation to fill this gap. So then we try. How we try this is we can generate a chemical recipe file, which is optimized condition for molecule scaffold. And this scaffold by principle, if you have a commercial catalog, it can give us like trillion size libraries. And this trillion size libraries we think is the synthesis ready library. That means once we have this protocol in the computer, we push the button with the platform, we should be able to make it. So then we collaborate with Prof. Deng in our School of Medicine. They are interested in this XCT target. And with this target, we actually also invited Prof. Chen Yuzong, who is an AI pharmacologist. Now he's actually moved to Tsinghua University. And he targeted on the similar targets use AlphaFold to do a transfer learning and focus on the XCT, do a focus learning, fine-tuning the AI model to generate a model. And with this model, we actually use a chop combine method to generate some molecules and then added the top 30 fragments in medicinal chemistry to modify those molecules. And then with those molecules generated by computer, the screen through all the drugability, like the principle, like toxicity, solubility, and then they use a machine learning model and also docking to rank those molecules. With the top 100 ranked computer molecules, we categorize them to 4 scaffolds. And then we think 12 of them we can make it by solid phase synthesis. And there's also eratosine and SAS, which is a benchmark molecule. But eratosine is very, very potent, but very toxic. And the SAS is not potent as enough. So far there is no, there is not a drug targeted on XCT. So my student actually developed these 2 molecules by solid phase synthesis. And each of them can actually be made by 24 hours. And this is a machine we built in the lab, and everything actually he bought from Taobao and built by himself. So it's pretty capable. And then we, with this scaffold, we actually do a long on-demand library, which is a synthesis-aware digital library. And we think we can generate both of them at a billion, at least more than billion scale. And then with that, we actually ranked the top 25 molecules and synthesized them. And you can see the black one, we synthesized them without problem, and the gray one is the one we filled. So it's like you do methodology study. Some works with the best condition, some may not. And another fragment, even more, we cannot make. But with the made one, we give to the computer, And, oh, sorry, we gave to Prof. Deng and get the biodata. And then we gave those data back to the AI or computer to refine the model, and then generate another refined model to do another prediction. So we generate more molecules based on the refined model. And then with all those together, we actually achieved two lead compounds from de novo design. This two lead compound actually have a potency between erastitin and SAS. But the good thing is that those two molecules are non-toxic at all. So we are further doing some mice study based on those molecules, based on those two lead molecules now. So this is our first try with AI integrated heat to lead optimization with automated synthesis. And it take us 3 years to finish those study. One thing is that it's the first time we do this, so it's slow. But another important thing is that we actually found the difficulty part is this chemical residue file generation, which take us more than 6 months. So this actually significantly slow down the DMTA cycle. So because there are inherent problems about solid phase synthesis, that is, we cannot monitor the reaction directly. And now we actually choose another method to solve this problem and can accelerate the chemical residue file within 2 weeks to speed up the generation. So now we can close the cycle more confidently. And based on those chemical residue files generated, we can generate a billion size of molecule scaffolds using both skeletal function and fragment functionalization. So hopefully we can accelerate the DMTA cycle. So with that, I would like to thank all my students. It's very hardworking to achieve some of our goal. And especially Chen Guang is the one who did the high-speed circulation flow. And the solid phase synthesis is also he pioneered that. Now he's working at the Fuzhou University, and Yan Bin and Duan Shuai are those ones who do the AI study. And Yang Bo and Jia Le is the one who developed the integrated universal flow system. And I also would like to thank my collaborators inside and outside the NUS, and also the funding agency from A-Star, GSK, Pfizer, and NUS, and the MOE of Singapore. And at the end, I would like to invite everyone to come to Singapore. It's a very small but a beautiful city. And it's on the equator, so every day is the same. And the leaves are always green all the time. And if you like durian, it's one of the best places for durian. You have more than 200 durian cans waiting for you to taste. So thanks again for being this kind of invitation. Thank you.
Thank you very much for Dr. Wu's excellent presentation. Actually, I was in Singapore for 3 years too. Okay, now welcome to Kejia. So, oh, sorry, introduction of your first. Okay, our next speaker is Kejia. So Dr. Ding is Executive Director of Chemistry at Viva Biotech. Dr. Ding oversees a portfolio of projects for medicinal chemistry and synthetic chemistry for global clients. He's one of our stellar employees for sure. As a distinguished leader of the Viva chemistry team since 2015, Dr. Ding has made a significant contribution to multiple chemistry projects that successfully reached the key milestones. He was loved by our clients, so very many successful deliveries. His core scientific expertise is in the medicinal and the synthetic chemistry for structure-based small molecule drug discoveries that's strengthened by his deep understanding of molecular structures and interactions. So medicinal chemistry not only make more molecules, you have to understand the logic behind that. He's one of our best. In recent years, he continues to lead chemistry efforts into developing new drug modalities such as cyclopeptides, bifunctional molecules, and various drug conjugates. Dr. Ding received his PhD degree in chemistry from UCSD, another very sunny place. And with his synthesis work focusing on design and the synthesis of small molecules targeting viral RNA. His work has been published in many journal articles and patents. We look forward to your presentation.
Thank you, Bing, for the very kind introduction. All right, so well, now we'll switch to the industrial perspective on the flow chemistry. And our thesis today is towards the automated synthesis in the lab in the loop AIDD. In today's presentation, we'll give a brief sort of sampling of very many cases from our labs and give you some understanding of what the traditional and practical applications of flow chemistry in organic synthesis look like in industry. And also, we're gonna touch briefly on the path towards the automated flow synthesis. At the end, we have some perspective on the automation and AI integration of various parts of the chemistry and also the engineering part. So Viva Chemistry is established back in 2009, many years ago, and we do have approximately right now like 500 bench chemists working across 4 different sites in China. We span from the small molecule sort of medchem projects, small molecule synthesis to all the way to commercial manufacturing. And our facilities are pretty up to date with all the modern semi-automated and fully automated instrumentations. Our synthetic capability spans from small molecule beyond Rudolph 5 compounds, so the larger sort of cyclic structures and including cyclic peptide and also linear peptide synthesis. Viva see its chemistry teams as an integrated part of its services. Now, more specifically, today we're going to look at the flow chemistry for organic synthesis. So typically, flow chemistry in a real setting is not a first choice for organic synthesis until now. So we typically apply it the flow chemistry because there is certain limitations in the traditional sort of solution-based synthesis. For example, when we need to run scale-up reactions, we wanted to achieve higher yield and we wanted to achieve some level of automation. When we deal with hazardous reagents, for example, sodium azide, some toxic or involving gas reagents. And also recently we've done a lot of photo and electrochemistry, and certainly I think running the flow chemistry, the solution through a tube, and then have the LED lights shining on it, does sort of provide a uniform light exposure, and so resulting in sort of more efficient irradiations and higher conversion rate and higher yield. We're going to talk a little about the solid phase synthesis, especially about the catalytic systems on the synthesis of small molecules and peptides. And towards the end, we want to show some of the progress and what we wanted to achieve in terms of a high level of automation and AI. So this is the standard sort of medchem lab flow chemistry instrumentation. And I think we also have now the auto having the auto sampler attached to these instruments as a potential run more automated process. But again, I just don't want to go into all the details. But again, currently in industry, such instrumentation allow us to run across a variety of chemical synthesis, many different types of reactions that was traditionally thought to be difficult. And also our CMC colleagues at Langhua Pharma, for a subsidiary of Viva Biotech, they can run kilogram and ton scale for a lot of the reactions that's considered fairly difficult for traditional sort of stir reactors. So next, I think that's the highlight, the main body of the presentation. We're going to look at the some cases from our labs in the at Viva Biotech. So the first compound is we are interested in this structure we highlight in blue, green. That's the cyclopentyl sort of benzofuran scaffold. Now this scaffold comes from a natural product, rocaglamide, and the synthesis of this compound was initially reported back in 1990, as we can see at the bottom left. that uses a oxidative cyclization of the dihydrofuran core to make the ring. And followed by 1991 report using a pedicle coupling of the aldehyde ketone. These early development of the synthetic methodologies for these compounds basically help confirm the structure of the compound, and it allowed the biological evaluation of the compound's activities and also the analogs. The more rational, however, the more rational proposal of the so-called biomimetic pathway, mechanistically speaking, is showing in the middle of this slide that should go through, presumably should go through a 1,4 addition and followed by the well-established keto rearrangement to make this core. However, this middle sort of initially proposed mechanism was never successfully practiced in the lab. Now, the major change for the synthesis of the compound or this core structure was reported in 2004 from Boston University Porco Lab, which also Dr. Bing was at back at the time. And so there is this selective photochemical 3+2 cycle addition reaction to make this scaffold. So we consider it a multi-step mechanistically speaking. So light was first of all involved at the first step, excited state intramolecular proton transfer to enable the formation of this short-lived transition state. Then can be cycle added to the other alkene partner through a 3+2 dipolar cycle addition. So once this bridged intermediate is made, then there is the well-known sort of keto rearrangement to form the desired core structure. Now, what's also nice about this chemistry is the there were a number of further development and optimizations based on this synthetic scheme. Later on, we have the citation down there. But again, this has been proven to be very, very reliable and very successful. Now, okay. All right, so that reaction was further developed into the flow chemical synthesis. Early examples from the 2012 JMC. This was actually an industry paper from Infinity Therapeutics, and on the right is the academic Porco Lab 2016 publication. So initially they decided to run this reaction in a sort of continuous recirculation flow setup. Again, mainly because you wanted to have more uniform and more effective light exposures to facilitate the activation of the core structure to form that transition state into the cycloaddition reaction. So the improvement was pretty good. So again, there is the throughput increase. Tens of grams of the compound now can be made in a few hours, as described by the authors, and they achieve 20% yield in 3 steps. On the right-hand side, there is the further improvement in the stereoselectivity toward to up to 5.1, and then with 46% yield. Now the setup of these photochemical synthesis is actually pretty straightforward and can be applied across most of the chemistry labs. So further development was reported in 2017 using a continuous flow setup for this particular reaction. So it's not sort of just from bottle A flow into the tube then back to bottle A. Now it's the setup that you only need to pass through the tubings once, and then from the other end you collect the product directly. So the scale up of this reaction was done by a serial sort of connection of multiple sort of light reactors, and and with a increase in the initial concentration and a changing of diameter of the tubings, etc. So this development has led to the throughput to 2 orders of magnitude greater than the corresponding batch reactors. So again, this is a scale-up application. Now, on the right-hand side, we show the Viva adaptation of this methodology. And first of all, what's different about for our compound is there is changes in the structure. So you cannot directly copy all the sort of methodologies from the literature papers, and you wanted to not in a metchem sense, you wanted to change your core, you wanted to change your substitution types, you wanted to change the position of your substitution. So there is further sort of optimizations and the tweakings of the flow setup and the conditions. So what we are able to achieve is first of all a very small footprint setup in the metchem lab hood, like we have seen in Dr. Wu's image, and we were able to achieve 20 grams per day throughput for the this is a fully formed scaffold and not only the intermediate. So, and then we achieve 40% yield over 3 steps. So the next example, we're going to look at a CH functionalization via the fluoroalkyl radical. Now, this reaction was the CH functionalization was pretty popular back in the time. The first publication that we always remember is the 2011 Nature paper by Macmillan, and using this trifluoro methyl sulfonyl chloride, under ruthenium catalytic conditions, form this trifluoro methyl radical, and then can do a direct CH functionalization on a variety of compound structures. And followed by the precedents, then we can see filbarine, this famous sort of a DFMS zinc reagent to make a more stable version of the initial reagent as a white powder, and then if you add initiators THP, and then you can again do CH functionalization. In the original paper, there was the heterocyclic expansions on the substrate scope. And of course, at the bottom piece, there is a visible light sort of catalyze the not only the radical addition to the microacceptor, but then followed by a intramolecular tandem radical cyclization. Now, all these chemistries are really cool, and then we start practicing these chemistries in the lab. And one of the problems that we encounter is the purchase of the starting reagent. Now, there are 2 versions of this. So we can see on the right-hand side example, the one is trifluoro version and the other one is the difluoro version. Now, there is significant difference in the chemical properties in these compounds. If you've done, metchem development, you realize these compounds actually, these functional groups actually perform very differently in the drug molecule. Now, one of the compound that we initially had a problem sourcing large quantity was the difluoro version. Now, so we had another look at this compound, this difluoro methyl sulfonyl chloride compound, and so we wanted to understand how it's made and why, and how can we sort of get more of it at a reasonable cost. So this compound turned out to be first reported in 1979, very early time. The formation was from this gas, sort of chlorodifluoromethane, R22, or I believe it's the compound also called a freon. And right now it's basically phased out, and you cannot have a very good sort of commercial source of this reagent. But the establishment of the synthetic pathway basically suggests that this compound is a carbene. It generates a carbene intermediate, then followed by the nucleophilic conjugate, the covalent bond formation. There have been, reportedly the further sort of development of such reagents that achieve the same purpose of generating the difluorocarbene. However, there is an associated cost with these reagents as well. So if you are trying to do a scale up and then on an industrial level, again, that you run into a problem about sourcing these chemicals and also how to control your budget. But these compounds still, present very interesting properties. Up on the bottom left, we show a 2017, a fairly recent paper, Nature Chemistry, to use the chlorodifluoromethane directly in a palladium catalyzed coupling reactions with boronic acid. So this was the flow case study that we showed today about how we attempted to make and successfully make this sulfonyl reagent, difluoromethyl sulfonyl reagent in our lab. And so the flow setup was shown here, and what we are, I think two key things that's difficult about this reaction if you run it in the flask. One is one of the reagents, the difluoro chloromethane, is actually a gas. And then the other thing is you need a heating of the reaction to make it work. So that's why the flow chemistry was brought to the practice. And then through, I think, a fairly short amount of time and optimization, we're able to set up this process, which then gives us very high throughput at 80 gram crude compound 3. That, by the way, that's the ethyl ether, not the thionyl chloride yet. But we're getting like 80 gram crude 3 per day, and then with a 85% crude yield. Now, once we get this compound 3, then the oxidative chlorination to make the thionyl chloride, to us, is pretty standard in the chemistry lab, and we can run a large quantity of that. So the next step, the next example that we show here is a multi-step continuous flow synthesis of this compound 5 that is a chemistry of Curtius rearrangement. Now, a lot of us run Curtius rearrangement in a flask, in a solution phase all the time. At a smaller scale, you don't realize that is a difficult reaction. It typically works very well. But at a larger scale, you run into problems very quickly. So first of all, there is handling of large quantity of sodium azide, which is a very toxic material. And then the other thing is in a Curtius rearrangement, you have the release of the nitrogen gas, and then you heat a reaction to certain degrees to make the rearrangement work. And the reaction itself is actually an exothermic reaction. So then in this case, when we need to run 1,000 kilogram scale, that's the output, by the way, project. So we adapt the flow chemistry again, and so the entire process development was done within 1 month, and the team initially did a sort of a stir reactor and the flow reactor comparison on a smaller scale with success, and then quickly adapted to hundreds of gram scale pilot demo within a week, and then followed by the full validation of the flow design and start running the manufacturing process within 2 to 4 weeks time. And so as they achieve the manufacturing of 1,000 kilogram in 2 months, basically, with a overall yield of 60%, and achieve 99% purity by HPLC. So this work is done by our CMC colleagues at the Langhua Farm. So here, the next example, we wanted to quickly bring up this packed bed reactors that allow us to run metal coupling reactions in the flow setup. So in this case, we show the solid phase, either the silica, the polypropylene, or the polystyrene beads or resin are functionalized with the phosphine, organic phosphine ligands attached to these resin, and then so they allow the palladium to be immobilized onto the solid support. So using these pre-packed bed reactors, we're able to run the reaction, for example, the Suzuki, and also on the right-hand side, we show a Sonogashira example. Now, in our experience, we know the first thing we notice is if you run not so large scale of a reaction, you actually achieve a much faster reaction time. So instead of stirring in the flask for a number of hours, you now can run it through once in a short amount of time, for example, 1.5 minutes on a milligram scale, and then achieve a much higher conversion and purity profile from these flow setups. Now, this we bring this up is because we wanted to show the correlation between, this is an example that actually Dr. Wu mentioned in his presentation as well. This was done in his lab, and the solid support synthesis of drug molecules. So now the difference from the previous example is right now the compound itself is immobilized onto the resin, and then you have multiple pass-throughs of different reagents and the setup conditions to have a complete sort of high-level automated control of the entire process. And so starting material in, and then immobilize, and then you have the product out from this setup. And we think this is actually very, very exciting because, again, it allows a very small footprint. And if we look at some of the details in the setup, and we quickly realize we can adapt it into a much larger operation scale in the industry. And also we can have multiple setups like this, and then to sort of greatly increase the throughput of our work. Now, the directly related chemistry for the solid phase synthesis is, again, it's peptide synthesis. Now, for natural amino acid or a limited number of increasing sort of a wider range of non-natural amino acid can now be done on the automated instruments. And we have a few of these in the lab, and they are routinely used to perform the fully automated synthesis. But there is a limitation to it. Once you go beyond the comfort zone or the boundary of what these machines can handle, under those pre-engineered conditions, then you have to rely on human beings to make more complicated molecules, and that which is typically the case, the bulk part of the work for any sort of peptide drug discovery program. So we still rely on the human beings to make these compounds by hand, by a manifold in the hood. So, and then now we start thinking, okay, if we can make these, for example, Dr. Wu's setup available to our peptide team, would it be possible for us to further improve our efficiency? And ideally, in the next step is can we fully automate some of the process and connect to the database so that our AI team can get involved and start optimizations like the one of the examples that Dr. Wu also showed. So the other case is certainly, so this is a case of the solid phase synthesis in the lab for a highly modified and is essentially a fully non-natural amino acid compounds. It's a cyclized version. So the technique that we use in the lab is a combination of solid phase synthesis and also solution based reactions. Again, right now, like there's so basically we take whatever that's available that works, and then to deliver the compound. So next, we're going to touch very briefly on the automation and AI part. So this is one of the early report on the automated sort of continuous flow end-to-end chemical synthesis, and they add the authors add a formulation part to their setup. So again, very compact. You can see in the image, a very compact system, and allow a multi-step continuous flow synthesis. And so starting material in, and then you have the end product out. Now, there are limitations to such systems, and we're fully aware, but we're not going to unfold it and then go into too much details. But such systems are available at a very early time, and since then we continue to see many of the development in a higher level of automation. So robotic chemists, we have the retro synthetic planning. We have the computer system, and also the more recently, I think it's the robotic flow synthesis by AI planning, the Coley Lab, MIT Coley Lab. And also at the bottom right, we have the closed loop reaction condition optimization case. So the way we see these is, again, it looks like there's certain level of integration into the design and the setup of all these systems. But we would like to take them apart and look at them as modularly, and then start thinking the way if we can take modules that we need and then fit into our ongoing wet lab chemistry and improve our efficiency, and to work with our AI scientists. So the ultimate goal is, again, to facilitate, to help and assist the development of the so-called lab-in-the-loop AIDD drug discovery process. That we have the AI sits at a central, and then have collaborations and support across a number of different functions, either predictive function or make things run more smoothly and more efficiently. And we're hoping that by doing so, we will be able to achieve the AI-driven, structure-based drug discovery platform, a full complete range of services to our clients, and speed up the drug discovery, and ultimately bring value to the patients that's in need. So that's my presentation today.
Okay, thank you very much for the excellent presentations from both speakers. Actually, it was such a unique blend of the academic innovation and the industry expertise. Now we'll move into the Q&A session. Please take a seat. So for the we'll address questions online first. Audience online, please keep submitting your questions you have. Then we'll answer these questions, and maybe we'll move to the in-person physically in this room. Okay, I do have a couple of questions. The question number one, they didn't mention the two, which one? So I guess both of you can take turns to answer this question. The question is, what is the difference between drug R&D with academia and the industry based on your experience? Maybe I'll start from Professor Wu first. What's the difference?
I would say there are many differences. So in academia, we, as a professor, I would say we have many functions. One thing is we want to push the boundary of chemistry. So everything we do, we aim for paper, we aim for novelty. We have to try something nobody have tried before. So that's our job. And at the same time, we have to make an education. We cannot let the student go too far away from which do something never been used for industry. So actually in academia, we hope we can have more connection or more discussion with industry to really see where the problem is. And yeah, we also hope we can apply our technology to industry more quickly, so convert them more quickly, especially nowadays AI is speeding up everything. Yeah.
So before you could I give you the real world word, the research? What is your guess? Because you never put your feet in the industry. What is your guess of the R&D industry? Or what's your feeling as a
my feeling in industry is everything is customer driven, right? It's money driven, or it's a real problem that really can. So no matter how beautiful paper we publish, how good method we publish, at the end is a function of the molecule that matters. As a synthetic chemist, because I have to say I don't know about function. So we rely on our collaborators, we rely on our industry partners, or we rely on the AI to tell us what's the function of the molecule. But maybe in the future, I don't know, the industry, and on the other hand, I really think the industry can change the world faster than us, because industry have more resource, can combine different teams to work together. As actually in academia, it's very difficult to find good collaborators. But industry, you naturally have this engine to put all the teams work together for one goal. And I think that's you can change the world much quicker than us. That's my feeling.
So I'll have the same challenge for Katja. So maybe you mentioned your understanding of of academia first, then give the answer of the industry experience, please.
Yeah, I want to say, well, first of all, so Viva Biotech is a drug discovery service provider, first of all. So that's a unique setting. It runs on its unique business model, and it covers a wide variety of services. So in this industry, I think the biggest difference is there are so many different types of chemistry that you need to understand and practice, and all these related problems you need to solve. That's quite different from the academia, where you tend to, as you know, an individual, you tend to focus on one particular or maybe two problems at a time. So in industry, the throughput is much higher, and you have a whole variety of chemical synthesis going in parallel at any time, and then you need to run between different types of chemistries and solve the problem very quickly. So at the system level, I think Viva Biotech is in a very good position in sort of integrating all these different functions into a systematic process, and with very high efficiency and a success rate for delivering the result. So that adds actually on top of the bench experiments and understanding of the science part to again, a whole new level that you have to have the ability to understand each other. For example, if we work with the AI team to develop a process, and you need to understand how that process is done from the computer end, and evaluate the output from the AI team for them to balance out the creativity from the AI program with practical aspects, and can you actually put in the lab. So again, there are limitations, there are advantages, running science in industry. I think it's quite different from the university lab, but so far I think I've had a very good experience and learning a lot of things from my colleagues.
Okay, great. I have the second question. Actually, it's addressed to Dr. Wu. So that's a very more detailed technology questions. So the question is, when the reaction mixture contains two immiscible solvents, like for example, water and the toluene, how can homogeneous distribution to be achieved in the pipeline?
So to answer this question, I think this question is asked for in the flow setup.
I guess so, yeah.
Yeah, so
didn't specify.
to mix them together, we actually have many ways to do that. If it's a homogeneous system, for example, there's no solid generation, we can achieve use a micro mixer, and normally that can help to mix in the two in like a insoluble mixture in a very uniform and very fine way. Use a micro mixer. That's my answer.
Sorry, how? That's very naive because I know it's like a water toluene.
So for example, you can depend on the normally the mixing efficiency in flow is quite good already.
Even like water and the benzene.
Yeah, you can just use the like the flow rate to control the ratio of these two reagents or these two solvents, and use a micro mixer, and they will mix very, very efficiently in so then the physically separate one portion to many portions, and then do the mixing by the micro mixer.
So okay, sorry, I'm also curious about this. So we'll make this solvent to very small bubbles, I guess, or very small particles.
Portions, yeah.
Portions, like, do you have an idea what's the size of these portions of the particles?
Oh, they can go like
nanometer
even scale.
So in that case, we'll solve this problem. Okay,
yeah,
great. Okay, learn something new today. Okay, the third question, they didn't address the specific to either of you, so I guess both of you will have the chance to answer. So the question that we just got is, will AI-driven, fully automated self-driving labs replace human chemists for synthesis? That's a kind of word from some nervous chemist, I guess. So from you, Dr. Wu. Oh, you did the work, yeah.
Sure, yeah. Well, as I described towards the end of the presentation, the way we look at this is we tend not to look at this thing as a one piece. We tend to break it down into individual modules and functions. So to answer that question, I think AI is probably not will, but already replacing certain tasks for to replace the chemist. But I don't really like the way to describe that as you're totally replaced. But I think it's just you let some of the tasks to be handled by AI or by automation. But when you gain access to those extra time, you don't need to spend in the lab anymore. Then we actually think that time is very, very valuable for chemists, so that you can have more sort of scientific thinkings and a design, and a better design of the chemistry. And then I think it's just it's not sort of you lose one function and then you don't gain nothing. So it's like you need to repurpose your workflow from more about handling these experiments into designing these experiments. And I think that's actually higher level requirements for existing chemists. So it's actually more demanding. But again, I think that's what technology development is about.
Yeah, I think adding on Dr. Dean's comments. So actually, I'm teaching advanced organic synthesis in NUS, and every time we will show the example of AI and automation to our students. And for example, Andrew Cooper's system, it's a robot, right? The robot can work like 24 hours per day, just need 2 hours to charging, and no need to breathe like oxygen. So they can actually really run the reaction in inert atmosphere to replace most of our students in the future. But now I think the progress is still stepping out because it's slow, right? But I think the world change faster than we think, at least than I think. Because like 10 years ago or 20 years ago, when nobody used smartphone, but nowadays the phone is like an organ in human world. So you never know how quick AI will develop. So what we can do is we only can focus on today, right? We don't know 10 years later how the world changes. Maybe like Elon Musk said, everyone no need to do job anymore. You just lay back at home and rely on the computer or robots. Everyone have 5 robots working for you. But that's a dream. But for now, I think as Dr. Dean mentioned, we may have higher requirement to students or to our chemists, like what's really differentiate you from you and at least the robot or AI. And we have to embrace AI. So AI will differentiate the people to even larger extent. The people or the scientists or the students who know how to use AI will be grow much faster compared to those ones reject AI. That's my thinking.
Thank you. Actually, I like to add one more thing. Actually, I was early trained as photo-organic chemist or photophysical organic chemist. So the very beginning, I thought it's kind of like not the hardcore synthetic group. I did natural synthesis. But now later in my career, I realized that the training of physical organic chemistry actually play a very key role here. It's like, understand the energy, the lifetime of the intermediates. Those very helpful for any downstream job like medicinal chemistry, the acid development stuff. I think echo the Dr. Wu's thinking, so the students or our younger generation chemists, you maybe the solid fundamental understanding will definitely help you to beat the AI or just the robots. That's I guess. Okay, one more question here. Actually, I think you guys will take turn to answer. So the question is, when you have a reaction, it's a new reaction to try, you always use flow chemists as the last straw, everything failed pretty much, or you'll give it a try at the very beginning.
So for my student, I think they will still try the reaction in batch, especially in GC valves. So if we can get every reaction in GC valves working, for our high-speed circulation flow, we can make them to large scale. So when we try reactions, we always want to try in a high throughput manner.
Yeah, I think in my world of chemistry, I think the default option is still the solution synthesis. And but again, I do see a clear trend of having more and more of the photo chemical reactions running in the lab. And then as you have more of such type of reactions, and again, the immediate thing that you wanted to have is to have these flow reactors, which again, as I mentioned, they greatly improve the efficiency of those photochemical reactions. So yeah, I think they just gonna keep coming, and then until to sort of rebalance. I think again, flow chemistry has a scope, and the solution chemistry has a scope too. So eventually they will reach a balance. So I don't think there is gonna be like a total turnover right away, but it also depends on the chemical technology that's behind for example, the catalyst development, some of the, I think the iterative CC bond formation that we saw. So all these things will sort of gradually change the landscape or the way how our chemists handle reactions.
Good.
I can add a little bit more on this question. So I think a batch system is very good to screening discontinuous variables like a catalyst, solvents, substrates. But continuous flow is very good for screening continuous variables, such as time, pressure, temperature, component portions. So it depends on what you want to optimize. You can choose different technologies. So normally we want to optimize the discontinuous variables followed by continuous variables. So when you go to continuous variables, I think a flow is a very good tool for screening.
Okay, great. Okay, I gonna take the privilege as a host to ask a question. So actually, it's such a small community. I just realized that's N version of the 3+2 reaction. Actually, I was the original one developed that, but I've lose track of that. Dr. Ding, so for the flow chemistry, how big the scale of that reaction is, like grams or 50 grams or the 3+2 N version?
I think in our operation, so the numbers that we report in its presentations were pretty much the numbers that we need, but I don't see there's a cap on that. So if I think we can definitely sort of further scale up or run multiple parallel flow setups if we are restricted in the MechChem space. Now, if we give that to our CMC colleagues, I'm sure they can run ton scale.
Wow. Good to know. Okay, I think this is relatively late for the online, especially our US colleague, our US friends. So we'll kind of close the online questions. We'll keep the questions discussion in physically in Shanghai a little bit longer. But while we wrap up, I'd like to thank the Viva Biotech sponsorship. Thank you very much for Viva Bio Insights give this opportunity to have the chance for everybody to share the love of chemistry of science. Honestly, that's a great opportunity. And of course, thank you very much, our two excellent speakers today for the terrific talks. And also thank the audience in this room, online, for their attention, for their engaging questions. Thank you for your time. And also thank you very much for your continuous support to the Viva Bio Insights. We're looking forward to the next event. Thank you very much.